ecology, by definition, this is the study of the relationships between living organisms. This includes humans and their physical environment. And it aims to comprehend, to grasp the vital links and bonds between plants and animals, as well as the world around them. In this laboratory course of ours, we tackled here the practicality of ecology itself in general, where we became ecologists for the course subject. We utilized here various software applications such as QGIS, SPSS, BioDiversity Pro, and Photoscape. We acquired enough knowledge needed on using the said software apps and applied it to the fieldwork that we have done within the campus. And details on what exactly those software apps are will be further explained by other members as we proceed more into the topics. In data management, we were given 9 subjects of male and female students. So we acquired here individually their height in centimeters of measurement, and we calculated for its t-test result. It was then computed to have a less than 0.05 result, which would mean that there is a significant difference in the calculations. For the fieldwork task, the campus was divided into quadrants, to which different groups were assigned to the different parts of said quadrant. So our group was given the area of the science building, the FEU Memorial Square, and more than half of the portion of the Pavilion 1. Our group spent days acquiring data on the areas, GPS, the highest and lowest altitude of the area and its description, the number of plants and trees as well as its average height, and the number and description of animals found within the vicinity.
There are various diversity indices that can be utilized to measure species diversity in a given community. First is Shannon-Wiener Index, denoted by H', is an information statistic index, which means it assumes all species are represented in a sample and that they are randomly sampled. Values for H' are within the range of 1.5 to 3.5. The higher the result, means higher species diversity. Simpson's Diversity Index, denoted by D', is a measure of diversity, evenness, and dominance, which takes into account the number of species present, as well as the relative abundance of each species. The value closer to 1.0 shows a higher diversity. Sorensen's community coefficient of similarity is used to determine what the communities have in common. It gives a value between 0 and 1. The closer the value is to 1, the more the communities have in common. Species identification activity involves 3 different species of trees, namely Nara, Banaba, and Acacia. To conduct identification of these trees, picture keys, such as those available in online resources, were utilized accordingly. Closed-up images of these trees, as well as their reproductive parts, were taken and collated. Shown in this file is a sample output for this activity.
Quadrant sampling is an important tool in order to quantify the relative abundance of organisms in an area. In this activity, we learn how to estimate the population of Nara by using the formula of N equals capital A over small letter a, where capital N is the estimated size of the entire population, capital A is the area of the entire population, small letter a is the area of sample size, and small letter n is the number of individuals in the sample area. First, we measured the area of the entire population by taking the length and width of the FEU campus, which was 34,340 meters squared. Next, we divide the whole area to 6 sampling points and divided each sampling point to 4 specific sampling points. Then, 10 random sampling points were chosen, and in each sampling points, there were 4 specific sampling points in which we measured a 25 meter squared area of sample size. If Nara was contained within the 25 meter squared area of sample size, it was counted and recorded. Overall, the number of individuals in the sample area was 0.9. The estimated size of the entire population of Nara was 1,236.24 Nara trees.
QGIS is a free software that can be used to create maps as well as to add data and customize the maps that are created. The activities we had with regards to QGIS map making is the creation of maps for FEU campuses, creating a map of our hometown, as well as a practical exam to check our mastery and knowledge in using the app. As third year and fourth year biology students, we are currently taking our thesis subject. One of the parts of our thesis paper is the collection site, in which we can use maps to help illustrate the collection site as well as to improve the appearance of the paper. As a biology student, one of the highlights for study is the fieldwork. In preparation for our supposed fieldwork, we were tasked to watch and transcribe videos to help us understand that there are a lot of studies that can be conducted in the field, as well as different ecosystems that can have varying factors and aspects to be learned and studied.
SPSS is statistical software commonly used in ecological research for analyzing and interpreting data. Ecological research often involves collecting large amounts of data on environmental variables, population dynamics, and species interaction. SPSS can be used to organize and analyze these datasets, allowing researchers to identify patterns, trends, and relationship between variables. BioDiversity Pro software can be useful for ecology students by allowing students to analyze and visualize biodiversity data like Shannon Index. With this, BioDiversity Pro can provide insights into ecological concepts and theories, while also helping students to develop the skills and experience needed for success in ecological research and related courses. Photoscape Splitter is a feature of the photo editing software Photoscape that allows users to split an image into several smaller pieces. Like we do in our activity, Photoscape Splitter allows users to specify the numbers of rows and columns to split the image into, as well as the size of each piece. Once the image has been split, users can edit each individual pieces separately or combine them into a single image again.